Hey everybody, it's time we should get to work here and talk about some Bio 1120 stuff. Here's the deal. In lab this week, you guys are going to be working on enzymes. Okay, you're going to be playing around with enzymes, looking at how they catalyze chemical reactions. Next week, you're going to be talking about, you'll be using microscopes to look at some pretty cool stuff. Pond scum, for instance, one of my all-time favorite things to do. I don't indulge myself enough in using a microscope to look at pond scum, so count on seeing me in lab next week. Guilty pleasure of mine will be, I'll be there trying to see some cool, weird things in the scum that you guys get to play with. But the lecture this week is going to be focusing on essentially the things you'll be talking about in lab and working with in lab next week. And the things that we'll be doing in class next week will be partly taking a test. Remember, that's coming up on Thursday. And then also a little bit talking about some of the enzyme kinetics that you'll be doing in lab this week. So bottom line, this is all me, I think we should have flipped the order of these two labs. That would have made some good sense. And so subsequent offerings of the class, I'm going to see if we can't have lab 4 come before lab 3. It just probably would mesh a little bit better. But so if you're wondering what's going on between the lab and lecture, they are linked. It's just we're kind of like in flipped order for the next 2 weeks. So that's what we're in for in lab and here in lecture for the next week or two. Before I get to the main topic at hand for today, though, there's one thing at least that it seems to me that was important that I neglected to say anything about at the end of the lecture on Thursday of last week. I was talking with you on Thursday about macromolecules. I was talking specifically about proteins and nucleic acids. And there's just like this profound concept that drives so much in biology that I didn't even mention, and I've been kicking myself waiting for a chance to tell you. And it turns out it's relevant to the lab that you're doing this week, so that's maybe my excuse for saying it now. But here's this concept: structure and function, right? Maybe just leave it just at that, just structure and function. You know, if we want to expand on it a little bit, I'd tell you structure and function in living systems at all levels, at the molecular level, at the cellular level, at the tissue level, at the organismal level, at the ecosystem level, at all levels, structure and function are intimately related. Okay? That's a universal truth for biological systems. And more surprisingly often, and I think I'm inclined to say always, if you have a good understanding of the structure of something, you also then can infer and make some pretty good guesses as to how it is that it works, how it is that it gets something done. And the reciprocal is true. If you know that something needs to be done, that usually informs you about what sort of structure would be necessary to do it. So you guys are sitting there thinking, what's he talking about? Structure and function. Okay, I got those words. I can link those 3 words together. But why it's relevant to what I was talking about on Thursday is pretty simply this. I should have drawn your attention when I was talking with you about primary and secondary and tertiary and quaternary structure of proteins. I kind of pointed to active sites of a couple enzymes, as I recall, right? But how would you change that structure? And if you changed that structure, realize that that would in turn change the function of the molecule. If you changed one of those 3-dimensional structures that I showed you such that the active site was closed instead of opened up as a gap, that's going to have a big effect on the ability of that enzyme to catalyze a reaction. Substrates won't be able to get in if you've closed off the active site. And then that in turn brings me to a neat sort of idea. How would nature change the structure of a protein once it's made the protein? Right? Appreciate why I said once it's made the protein, because you could change the structure of a protein by assembling it with a different sequence of amino acids. That's one way you could change the structure in some sense. Living things don't do that. what's spelled out in our genes is what comes out as the final version of the protein. But conceivably, you could make a protein with a different structure by stringing together different amino acids. But my question right now is you've made a protein with a particular sequence of amino acids that's folded up into a particular secondary structure of alpha helices and beta sheets, and that in turn has given rise to a three-dimensional or a tertiary structure. How do you change that tertiary structure? That's a beautiful question. I love that question because it's going to get you thinking about a lot of the stuff that I hope I've been trying to impress upon you for the last couple of weeks. You could change that structure in a bunch of different ways. One of the ways you could change the structure is changing the pH of the environment in which that protein is hanging out. If you increased the hydrogen ion concentration, so if you lowered the pH, that's going to have an effect on the charge of some of the amino acid R groups that were deployed on the surface of the protein. Some of those that had a positive charge, so let's do it the other way, some of those that had a negative charge will now be picking up a hydrogen, and that negative charge is going to go away, right? A carboxyl group that now takes on a hydrogen because there's a whole bunch of excess hydrogen in the solution isn't going to have a negative charge anymore. If it doesn't have a negative charge anymore, it might not be as hydrophilic. It might not have the same affinity for an amine group somewhere else. The amine group is probably going to be more likely to have a positive charge. Right? And so you're going to be playing games with the deployment of charges in hydrophobic and hydrophilic patches on the surface of the protein. And I hope then it's really easy to imagine how that's going to cause some parts of the protein to move away from where they were before and to interact with other parts. You can change the structure of a protein by changing the pH, the environment in which it's hanging out. You can have lower pH, you can have higher pH, they're going to have different effects. You should expect that changing pH in either direction will change the structure of a protein. And coming back to where I started with all this, if you change the structure of a protein, you should fully expect that you will be changing the function of that protein as well. Okay, so you can control the function. You can turn you can turn a protein on, and you can turn a protein off by playing games with the pH in which it's hanging out. How about that? How else could you change the structure / function of a protein? pH, that's an easy one, I hope. How about temperature? If you increase the temperature, things that were kind of lethargic and hanging close to the main body of the protein may now start to get wiggling around and dangling out and might be doing something different. If you lower the temperature, things that were dangling out may just kind of get stuck in place as opposed to feeling around the way they were before. So changing pH, changing temperature, those things are quickly going to change. You should expect, not always, but you should expect that they will change the function of any given protein. You know, when you get a fever, right, there's some fevers you just don't worry about, right? Like 100 degrees Fahrenheit, 101 degrees, not that big a deal. You know what's going on there? It turns out that through natural selection, your body is a well-tuned machine. Your body can tolerate those little increases of temperature better than the things that are infecting you. Right? And so you increase your temperature, makes you feel a little bit loogie for a little while, but the bacteria, typically, where the viruses that are infecting you, they take it on the chin because they're working in a narrower temperature range, perhaps, than you are. So it's a good strategy. But at some point, if your body temperature gets too high, it starts to cause problems. Usually at around 104 or 105, you should start getting worried. At 105.5 and beyond, at that temperature, disulfide bonds get broken, like the bonds that get made that we were talking about between cysteines that staple together different parts of proteins, right? At 105.5 and beyond, those bonds get broken, and once those are broken, they can't be remade. right? Those get made as the protein is being freshly minted in certain compartments in the cell, which we'll talk about later today. You pass through that compartment once as a protein, you never go back, and so if that bond gets broken, that is now a non-functional protein. Did you ever consider what happens to egg ovum albumin, the egg white protein, right? When you cook an egg, that's why we call it an egg white. What you're doing is you're denaturing the disulfide bonds in egg ovalbumin, and those can't be brought back together. There was an Ig Nobel Prize that just got awarded to somebody who tried to reverse that process. It doesn't work. If you know about the Ig Nobel Prizes, you'll appreciate what I've just said. If you don't know what I'm talking about, don't worry about it. It's not going to be on any test. All right? So, how do you change the structure and then the function of a protein? You can play with the pH. You can play with the temperature. There's one last way that I want to really make sure I draw your attention to, because in lab this week, those are the two things that you're going to be playing with, because those are easy for us to manipulate in a lab setting. Something that's not easy for us to manipulate in a lab setting, but nonetheless is a real go-to move for living things, is by tacking things on or removing things from the surface of proteins. There's a thing called phosphorylation, right, where an enzyme can put a phosphate group on the surface of specific proteins. Can you imagine what that does to a protein's structure? If somehow or other parachuted in is a little intense cluster of negative charge somewhere on the surface of the protein. Oh my gosh, that's gotta freak the protein out. There's gotta be some response to that negative charge. If you put a phosphate group on, all right, what's going to happen is that's going to change the structure of the protein. That's going to change the function of the protein. Sometimes putting that phosphate group on will inactivate an enzyme. It'll change the structure in a way that stops the enzyme from doing its thing. Sometimes it activates the protein. It changes the structure that now the active site is open. Right? Sometimes you need to remove the phosphate group to get the protein to work. Sometimes you need to put the phosphate group on. The point here is that dropping on a local negative charge should change the structure of most proteins, and when you change the structure of a protein, you typically change its function. So you can put phosphate groups on. Proteins are also often methylated, so a methyl group will get dropped down to them. There are those sorts of manipulations that cells use all the time to regulate the activity of their enzymes, and they do that again by playing with this really neat fundamental concept that structure and function are intimately related for living systems. And the first whiff of that we should have gotten if I was on my game on Thursday, the first whiff of that we've gotten is at a very molecular level talking about the structure and the activity of proteins. Today, now, I want to start talking about more complicated structures. Even than proteins and nucleic acids and carbohydrates and lipids, I want to talk about what living things do once they put all that stuff together. For the first time today, we're going to be talking about things that are unequivocally biological. All right? We're clearly in the realm of biology, because today I want to talk with you about what cells look like. All right? And bear in mind, as I'm talking with you about what cells look like, that I'm going to be talking with you about structures. All the while, in the background, in the back of your head, think to yourself, how is it that this structure has something to do with function? How does this structure impart some functionality? And I can tell you this very honestly. If you don't know why a particular structure is there, that means you've missed out on some important function. Okay? Everything that we're going to be talking with you about, nature is very parsimonious. It doesn't have stuff that it doesn't need, right? Especially elaborate, fancy structures. If there's a structure that we're talking about, it's there for a functional reason. And if the cell can't execute that function, it can't compete effectively with other cells that do, and it won't hang around very long, right? That's natural selection working for you when that sort of thing happens. So today I want to talk with you about cellular structures. as I'm doing that, if I don't make it clear, we got to share the workload here a little bit. I want you guys to be wrestling, trying to figure out what those structures do from a functional perspective. I'll try and spell it out to you, but if I drop the ball, I need you guys to help me and pick it up and carry it a little bit. Okay? So, in lab this week, you're going to be playing mind games on proteins, structures. You're going to be changing pH, you're going to be changing temperature, and you're going to be looking to see what effect that has on the activity, the functionality of proteins. In lecture today, let's talk about the structure of cells themselves. So, let's start by drawing your attention to this idea. Well, even before we get to that, let's talk about the scale of things a little bit. It was Anton van Leeuwenhoek who made the first microscope back in the 1600s. Before that, nobody had any clue about any of these cellular structures that we'll be talking about today. It wasn't possible to talk about structures until you had the advantage of a microscope. The short reason, the bottom line for that is that cells tend to be too small for us to be able to see with our naked eye. With really good eyesight, people with the very best of eyesight can discern the difference between something that's 1 point and 2 points if the 2 points are 2 millimeters apart. Anything less than 2 millimeters apart, nobody has visual acuity. No human has enough visual acuity to be able to discern that, hey, those really are two separate spots. So anything smaller than 2 millimeters is just not going to be something that you could get a look at unless you have the assistance of something like a light microscope. If you look at the scale of things here, 1 millimeter, right, is right at the edge of where we start to get help from light microscopy. What kind of things are bigger than a millimeter? Well, look at that. There's a cell you could see with your naked eye, a frog egg. Some of you, many of you probably have seen them if you've spent any time playing around in ponds. Chicken eggs are something you can see. Right? How about that? That's a neat example of a mammoth cell. In human beings, they're not cells, there are a bunch of cells, but right, you know, you can see. So these are the sorts of things we can see with our eyes. We're not seeing a lot here in the way of actual cells, and you need light microscopes to be able to get into that realm. But boy, right in smack dab in the sweet spot of what light microscopes can show us are things like plant and animal cells, and even things like mitochondria. That's supposed to be a mitochondria there and there. We'll see better pictures of them in a little bit. And also prokaryotic cells. Okay, so in 2 weeks in lab, you're going to be looking at a bunch of prokaryotic cells, and there'll probably be a few eukaryotic ones that you'll see swimming around as well. Okay, so that's the kind of scale that we're talking about here. Prokaryotic cells tend to be about 1/10 the size of eukaryotic cells. And viruses are way at the lower end in terms of what it is you might hopefully be able to see with a light microscope. And to really get a close look at things, you need to start invoking electron microscopes. That gets you a glimpse at things about 100 to 1,000-fold smaller even than a virus. But even still, at the end of the day, it's a real stretch to be able to see proteins with any methodology, and molecules like water, geez, you know, that's just sort of a conceptual thing. Nobody actually has been able to use an instrument to look at a water molecule. And things like atoms and subatomic particles, again, that's just all in somebody's imagination. Okay? So in terms of what it is you might possibly be able to see, what you guys are going to be working with in the lab are light microscopes. And that's going to be pretty much what we'll be talking about today. These are the sorts of things that Van Leeuwenhoek could have seen and in some instances did see. Mixed in with all that I've just rambled about, there were a couple of things that might be worth remembering. The relative size of prokaryotic and eukaryotic cells. Prokaryotic cells tend to be smaller than eukaryotic cells, about 1/10 to 1/100th the size of a typical eukaryotic cell. Right? That's one sort of vague idea that might be useful. And you really have very little prospect of being able to see proteins even with an electron microscope. But some assemblages of proteins, like the ribosomes, which we'll be talking about later, do get to the point where you can see them with a light microscope. So we'll be taking a look at those a little bit later today. So there's some sense of scaling for you. Let me take a few minutes now to just say a few words about prokaryotes versus eukaryotes. Let's start with the breakdown of the word, the origin of the word. Eukaryote, okay? Eu is Greek for with. It means it has something with something. Karyo, in this context, is derived from the Greek word that's equivalent to kernel. You would sound a little bit alike, right? And so eukaryotes are organisms with a kernel. That's what the name is derived from. The kernel that we're referring to here is the nucleus. Okay? So organisms that have nuclei, the plural for nucleus, are eukaryotic organisms. Organisms that do not have nuclei, with is eu, without is pro, right? Organisms without a nucleus are prokaryotes. And so what we've got here is an interesting way of dividing, parsing out living things, splitting them into different categories. We have one category where we talk about organisms that have nuclei, and we have another category where we talk about organisms that do not have nuclei. As a general rule, categorizing things on the basis of not having a feature, like a nucleus, is a really ineffective, bad strategy. For every rule, there are exceptions. It turns out this looks like it's a pretty good exception to that rule. Dividing living things, parsing them in that way, seems to be a pretty useful way for us to think about living things. With that said, right, this is a fundamental split in living things, an ancient evolutionary split in living things. The origin of nucleated cells as opposed to non-nucleated cells, that was an important event in the history of the evolution of life. It's ancient. It's like 1 billion years old. It's been a long time thing that this has been going on. With that said, I want to make sure that I at least say something about the fact that prokaryotes are not the only game in town in the sense of things that have a nucleus. There's another class of organisms that we call the archaebacteria that are just as anciently split off from the sort of root stock of life as eukaryotes and prokaryotes are. So archaebacteria are as different from prokaryotes as prokaryotes are from eukaryotes, and as eukaryotes are from archaebacteria. This is a very ancient split that gave rise to the archaebacteria relative to the prokaryotes, and also the split that gave rise to the eukaryotes from probably the prokaryotic lineage. Okay? Archaebacteria are things that it's very unlikely you will encounter at any point in the course of your life. They haven't fared quite so well in terms of conquering the world as prokaryotes and eukaryotes are, but boy, there are a whole bunch of them, and it's a very diverse population of things. They tend to live in exotic places like deep sea vents and geysers and stuff like that. So there's another class of life that I want to make sure you guys at least have had some exposure to. But most of what it is we know about how it is that cells work and how it is that enzymes work, we know from experiments that have been done on prokaryotes. Okay? Nobody gets up in arms when you talk about mass murder of billions of E. coli cells, right? In fact, a lot of people will pat you on the back if you come up with a way to kill 1 million E. coli cells, right? Because, you guys know what E. coli is, right? It's Escherichia coli, E amongst friends. It's a bacteria. It's essentially the lab rat of biologists who work on microorganisms. is a bacteria that normally lives in our poop. All right, so that's where you typically find E. coli. And there are some bad, mean E. coli that sometimes get people sick when they get out of where it is they're supposed to be hanging out and infect you in places that you don't want to have that kind of bacteria living. So nobody seems to mind. I've yet to encounter anybody who gets concerned when we talk about experimenting or manipulating or killing or playing mind games on E. coli or other bacteria. And as a result of that, that's where people focus a lot of their attention. They create mutant E. coli. It's a very common strategy amongst biologists. How does this system work? I know what I'll do. I'll knock out this protein. I'll take it out of the cell's repertoire. It won't be part of its arsenal anymore. And then I'll look to see how it's got screwed up. What is it that these messed up bacteria can't do that non-messed up versions can do? So experiments on prokaryotes turn out to be the primary way that we learn how enzymes and other cell structures work in all living things, archaebacterial prokaryotes and eukaryotes alike, just because there are no significant prohibitions about what you can do to a prokaryotic organism. Eukaryotes, however, are what we are ultimately probably the most interested in, because if it hasn't become clear, we're eukaryotic organisms. You know, what happens for a prokaryote is all well and good, but we really want to know how to improve the human condition, how to cure human diseases, what our capabilities are. Maybe we have capabilities we don't realize that we have. So ultimately, we want to study eukaryotic organisms. Most of what we know about eukaryotes we've learned from beating up on poor little prokaryotic organisms. Okay, they grow fast, they don't make noise when you hurt them, and you know, they're good, simple model organisms. So with that said, let's take a moment to pay some respects to a prokaryotic organism. A couple pictures for you here just to get us conceptually talking about what prokaryotes look like. On the left side of the screen here is an artist's rendering of a cell with pretty colors, and on the right side is actually an electron microscope image of a cell. So that's the ground truth on the right. The left is an artist's rendering to help you notice the more important details of the sort of thing you might see in the real world on the right. So you can see a bunch of stuff has been labeled in this particular cell. They're calling it a typical rod-shaped bacterium. They're giving you the specific species of it there on the right. That's a Bacillus coagulans. I've never heard of that species before, right? There are lots of species of prokaryotes, not a big deal. But the point here is that's fairly typical. You know what I see when I look at these pictures? I'll tell you. I'm a pretty honest guy, right? I'm not hiding any punches from you guys here. What I see when I look at the thing on the left, I see a bag of goo, right? That's a pretty good working operational model as to what a prokaryotic organism is. It's a bag filled with gooey stuff, right? Let's be a little bit more formal and scientific about this. The bag has a fancy name. It's a plasma membrane, right? So it's got to be up there somewhere. Look at that. Right in the middle of the screen, there's a line that's pointing to the bag. The bag has a name. The bag's name is a plasma membrane, right? You know, if you go to if you're a physician someday and you're talking with a patient, you know, we're going to give you an antibiotic that is going to break up the bag. That's the bag of goo. It doesn't sound nearly so good as if you say, we're going to give you an antibiotic that's going to disrupt the plasma membrane of the bacteria that's infecting you. Right? So this is where you learn to say those sorts of things, as opposed to talking about the bag of goo. But between you and me, that's a bag, right? The plasma membrane is a bag. And it's defining the boundary of the cell. We've talked about this in the very first lecture of the class. There's something that's outside the cell, as opposed to stuff that's inside the cell. The boundary is that plasma membrane. And in lecture a week ago today, I showed you what that plasma membrane looks like at a molecular level. It's a phospholipid bilayer. So you've got two of those phospholipids tail to tail with the phosphate groups pointing to the outside of the cell and another phosphate group pointing to the inside of the cell, going all the way around. That's what the bag, the plasma membrane, gets made from. Phospholipid bilayer. So you got a bag, all right? Whoa, sorry about that. Bag, a very good place to start. Let's talk a little bit about the goo. We're in the home stretch talking about prokaryotes. There's not much to them. They're simple. What's the goo? Well, there's one prominent thing in the goo, at least in the artist's rendering, because apparently this artist has bought into the idea that this is important and we got to make sure that it pops out at you because it's really important. The thing that kind of pops out at you, I hope, is that purplish thing with the name bacterial chromosome. So inside of this bag of goo, there's a big molecule, a large nucleic acid, a long polymer of nucleotides that is the bacteria's chromosome. Bacterial chromosomes tend to be circular, so big circular molecules, no beginning, no end, just big circle. And when I was taking a class like this, I was told emphatically bacteria have one chromosome, period. Well, it turns out we know a little bit better now. Many bacteria have more than one chromosome, not just copies of a chromosome, but they'll have a chromosome 1 and a chromosome 2. There will be more than one chromosome in there. But when you look at that picture on the left, you can tell that the artist here really wants to make sure you realize that's an important, it's a big molecule, and it takes up a pretty big chunk of the real estate inside of the bag that is the plasma membrane. And that's a fairly reasonable representation of what's going on. You know, if you look to the electron microscope image to the right, TEM is transmission electron microscopy, if you're curious. If you look to the electron microscope look at the right, you can sort of see there's no line pointing to it, but you can kind of see squiggly things, right? Like this stuff over here. Notice how the bacterial chromosome is being pointed to over here. Plasma membrane, you point to it here, you point to it here. Cell wall, you point to it here, you point to it here. They don't bother pointing to the bacterial chromosome, but that's probably where the chromosome is. That's probably what you're seeing in those squiggles there. So the bacterial chromosome, important part of the goo. What else is in the goo? Well, let's start with some easy things. There's going to be a bunch of water in there. It's a watery environment, right? That's by far the easiest thing that one might be able to say. What else might be inside of that goo? What might the goo be made out of? Well, you know, the picture's not really helping you, is it? It's not giving you, all it's telling you is there's a chromosome in there. What else might be in there? Well, there's going to be a bunch of proteins, okay? Because the cell's going to be making proteins to do the stuff that the cell needs to do. Among the things that the cell needs to do is metabolize sugars, build sugars up, break sugars down. So there's probably going to be some sugary things in there. There'll probably be some things like RNA molecules that are conveying messages from the DNA to the ribosomes that are ultimately going to make the proteins. Ribosomes are proteins themselves, and in a little bit we'll talk more about ribosomes, but ribosomes will be in there making proteins, doing the things that the cell needs to do. Bottom line, it's a pretty good assessment to talk about it being goo. Okay? There's a bunch of stuff that's floating around. It'll differ a little bit from one cell to another, but the concentration of things inside the cell, inside the goo, as opposed to outside the cell is typically higher. It's almost always higher for things like sugars and proteins and amino acids and nucleic acids. Again, there's stuff in there. And beyond that, we probably don't need to get into much more detail. Right? There's just stuff in there, at least for a class like this. So there's a prokaryote for you. I think it's not at all unreasonable to simply describe it as a bag of goo. Very simple structure. There's not a lot going on when we're talking about prokaryotic organisms. You can start talking about things like these little hair-like fimbria. You can talk about the cell wall, which gives some structure to the just outside the plasma membrane. And for some bacteria, apparently not bacillus coagulans, but for some bacteria they'll have these little motors that are called flagella. Bottom line, prokaryotes are simple. Simple is good, right? It's not just a Quaker thing, right? Simple is good because it's hard to mess it up. It's a robust system. If you can make something simple that gets a job done, it's probably better than a complicated thing that gets the same job done. But more importantly for us, simple means it's pretty easy for us to understand and for us to manipulate experimentally. So here is a picture of simplicity. Now there is a picture of complexity. The previous slide was showing you a prokaryote or an artist's rendering of a prokaryote. How sophisticated do you need to be to look at this and say, okay, this is a lot more complicated than what was going on in the previous structure? I don't think you need me to explain that to you, but I'll tell you, whereas prokaryotes are simple, eukaryote, that's the word for prokaryotes, prokaryote equals simple, eukaryote equals complex. There's lots of stuff going on inside of a eukaryotic cell. And a lot of the stuff going on inside a eukaryotic cell has a special name because it's got a special structure, and that in turn gives it a special function. The big difference, the single big difference between prokaryotes and eukaryotes, it's not so much that eukaryotes have a nucleus, that's part of it. It's that eukaryotes have, write this part down, internal membrane-bound compartments. That's the big difference between prokaryotes and eukaryotes. Internal membrane-bound compartments. Among those internal membrane-bound compartments is the nucleus. All right? So that's part of the operational definition of a prokaryote relative to a eukaryote. Eukaryotes have a nucleus. That's an internal membrane-bound compartment. But while I'm on the topic of internal membrane-bound compartments, let me tell you what that translates to operationally, functionally. The advantage that you get from this complexity, the internal membrane-bound compartments, is this, is within a eukaryotic cell, within a single cell, you can have a wide variety of different chemical environments. Prokaryotes can't pull this off. All prokaryotic proteins have to work within the same fairly narrow range of pH and concentration of salts, sugars, and temperature. That's not the case for eukaryotes. You and I, within our cells, can have little pockets, internal membrane-bound compartments, where inside of that compartment, the pH is radically different relative to another compartment. Different in a way that would be utterly incompatible with the activities that are going on in other compartments, and vice versa. So we can play games. Because of this complexity, we can set up little micro-environments inside of our cell to game the system and take advantage of differences in pH, probably first and foremost, but also differences in terms of the concentrations of proteins and other things like sugars and salts and all the rest. So we can take advantage of those differences and have things that couldn't be happening simultaneously taking place relatively close by simply because they're in different compartments. And so when you look at a eukaryotic cell, the word complex jumps quickly to mind, right? But what's making it complex, again, ultimately, are all of these internal membrane-bound compartments. So this is what a eukaryotic cell looks like, at least from an artist's perspective. Dominating the field of view is the nucleus. It's perhaps the biggest. It's usually the most obvious internal membrane-bound compartment. When you see eukaryotic cells with a microscope a week from now in lab, the first thing that'll jump out at you, the way that you'll probably recognize it as a eukaryote, will be, hey, what's that big dark thing in the middle of the cell? Oh, it's got a name. It's called the nucleus. If you've got a really good look at a eukaryotic cell, you'll see within that dark spot that is the nucleus, a darker spot, let me try that a third time, a darker spot still that's called the nucleolus. there are different things that are happening inside the nucleolus relative to the rest of the nucleus. We'll talk more about that later in this class. But the nucleolus is inside of the nucleus. And then there are a bunch of other things. And we're going to go step by step through this cell. We'll peel away layer upon layer. We'll start at the kernel, the nucleus, and work our way out. But for an overview of the whole thing, you've got your nucleus, you've got things like the endoplasmic reticulum, the rough and the smooth. You've got things like the Golgi apparatus. They're even drawing your attention to these little bumps here, those little specks, which they're calling ribosomes. There are other things like mitochondria. So they're showing you pictures of 3 mitochondria. And I suppose in the mix, between the internal membrane-bound compartments, is cytoplasm, fancy talk for goo. Okay? So that's the lay of the land for a eukaryotic cell. Now let's take these one at a time and march our way through and talk about what each of these compartments is good for. Before I do, I feel strongly motivated to mention this. This is an artist's rendering, all right? You got to bear in mind the person who drew this was trying to attach significance to certain things in a way that was probably going to be understandable to you. 2 things. First, realize that when you look at a cell with a microscope, either an electron microscope or a light microscope, you're getting a look through a slice of the cell. Right? You can't see in 3 dimensions when you're using a microscope or an electron microscope. You see a 2-dimensional snapshot of what's going on inside of the cell. I mentioned it in the lecture notes. Let me mention it to you right now. There's a reasonably good chance that there's stuff going on in 3 dimensions inside of cells, associations, connections, interactions, that we haven't quite figured out yet because we're only seeing everything in a 2-dimensional sort of slice through the middle of the cell. And so this picture is kind of trying to give you the idea of 3 dimensions. That's just an extrapolation based on what people have figured out from 2-dimensional looks. And then the second thing, it really upset me when I figured this out about 10 years ago. How many mitochondria are in that cell? 3 by my counting. These orange things down here at the bottom are supposed to be mitochondria. So 1, 2, 3. Okay? How many mitochondria do you think there are in a typical eukaryotic cell? How about in a typical human cell? You know, based on pictures like this, I lived for something like 40 years thinking that there were 2 or 3 mitochondria in every cell. Guess what? The typical human cell has between 1 and 10,000 mitochondria. Right? What's up with that? 3 is a rough approximation of 1,000? What? Whoa, where did that come from? So I think that tells us something important about these pictures. Don't take them as gospel truth, right? They're there as a guide to help you appreciate the relative structures and some of the interactions that are going on here. But to put 1,000 little mitochondria in there, if they drew it true to life, you wouldn't notice. In fact, there's fewer ribosomes in there, the dots that they've got, than mitochondria. So they're smaller than relative to the size of the nucleus that what you're seeing here, and there's a whole bunch more of them. And all I know is that the number of mitochondria is way off in this picture. For all I know, there's similar problems. I'm not this kind of biologist. That's not my special area of expertise. For all I know, they may be off the mark on some other things. There's one nucleus. I know that. I've seen those, right? Mitochondria are really hard to see with a microscope. I doubt any of you will see them in lab next week. But bear in mind that sometimes the artists take a little bit of license with drawing these pictures. So for whatever it's worth, I toss that out to you. You know how I came upon the idea that there's typically more than 1,000 mitochondria? It's because I do get involved in court cases where DNA evidence is involved. A lot of times tests are done these days on mitochondrial DNA. And I'm scratching my head, how can mitochondrial DNA be so much more sensitive than nuclear DNA tests? It turns out it's because there's thousands of copies of the mitochondrial DNA inside of each cell, and there's just 2 copies of the nuclear DNA, one from your mother, one from your father. And once I had that sort of insight, I was like, oh, wait a minute here, this picture is just plain wrong, at least in regard to how many mitochondria there are. All right, so enough said about the artistic license that's been taken here. That was a picture of an animal cell. Here's a different kind of eukaryotic cell. This is a plant eukaryotic cell. The biggest difference between plant and animal eukaryotic cells is plant cells almost always have a cell wall associated with them. The cell wall can be anywhere from 10 to 1,000 times thicker than the plasma membrane. So for a tree's cell wall, that's the bulk of what you would see if you were looking at a tree cell, like in its trunk. It's almost all cell wall. Just a little teeny tiny bit of the goo on the inside. So the cell wall for plant cells can be a pretty big deal. But the basic nuts and bolts of what's inside, in terms of these internal membrane-bound compartments, is the same between both between both a plant and an animal cell. You got your nucleus, you've got your endoplasmic reticulum, you got this big thing here they're drawing your attention to called a central vacuole. Plant cells will sometimes store water here, sometimes they'll store starch here, sort of set aside from the rest of the cell. And then another big difference, oh yeah, how many mitochondria are there? 1, right? Again, artistic license. How many chloroplasts are in this cell? Well, this is a pretty unusual plant cell to have only 3 chloroplasts. There probably are such plants, but just as with mitochondria, these chloroplasts, those are uniquely present in plant cells relative to animal cells. And just as with mitochondria, a typical plant cell will have many hundreds or thousands of chloroplasts. Here they're just showing you 3 so that you can get a sense of the pretty internal structures within them. So we've seen pictures now of 2 different kinds of animal cells. Bottom line, 2 different kinds of eukaryotic cells. Bottom line, eukaryotic cells are complicated. What makes them complicated are internal membrane-bound compartments. And here again is a prokaryotic cell, simple. Biggest practical difference between a prokaryotic and eukaryotic cell is the prokaryote has no internal membrane-bound compartments. The front end of the cell has the same chemistry going on as the back end or the middle or the top or the bottom of the cell. That's not the case for eukaryotes. Okay, so let's get to work and talk about some of these compartments and what they're good for. We can start here with, whoa, how did that happen? Oh, let's talk about cell walls and membranes. All right, so working our way from the outside in, let's talk here about membranes. Phospholipid bilayer. You've seen a picture similar to this already in this class. Start with the pretty easy, familiar stuff. That is a phosphate group, or at least an artist's rendering of a phosphate group. The 2 spindly things dangling off of it are lipids. That's a phospholipid bilayer, by 2. All right? So there's your phospholipid bilayer. When you look at this, put on your chemistry goggles here and tell me functionally what function will that structure dictate? What functionality do you get from a structure like a phospholipid bilayer? I'll speak for you. It'll be more convenient and efficient that way. There's going to be a real affinity for water out here. This surface is going to be very water soluble. there's going to be a real affinity for water down here. The inside of the cell is going to have a nice, happy, watery environment that's going to love interacting with those phosphate groups. The outside is set up with the expectation that the rest of the world is going to be watery. That's the basic design that's going on here. So the outer edge and the inner edge of the membrane is designed in a way that makes it look like it's geared up to work with water on the outside and water on the inside. But, and there's a but, you got this lipidy stuff in between, this very hydrophobic stuff in between. This stuff that water won't want to have anything at all to do with. Water on the outside of a cell has a really hard time getting to the inside of a cell, and vice versa. Water on the inside doesn't get out very easily. For a water molecule to get from outside to in, it's got to fight its way through this very uncomfortable environment for water. And it's so much easier for once it gets a whiff of that to just bolt and run and go back to its other watery friends, as opposed to going through the no man, or better, the no water man land that is the lipidy part of the phospholipid bilayer. Right? And so this membrane is pretty impermeable to water and things that are water soluble. All right? Now, you guys have noticed there's more in this picture than just the phospholipids. There are those blobby things that have the name protein attached to them. And you guys certainly know enough about proteins now to appreciate that some proteins can be designed, can be put together in such a way that they have on their surface R groups that are hydrophobic and cause them to be really comfortable, perhaps only comfortable, when they're hanging out in the lipidy part of the phospholipid bilayer. On Tuesday of next week, maybe a little bit on Thursday of this week, I'm going to talk with you about phospholipid bilayers and how it is we know what we know about them. But those proteins are called integral membrane proteins. They're integral parts of the membrane because they have R groups on the surfaces of these regions of the protein and through here that are fat soluble but not water soluble. And many of these membrane-bound proteins will have some portions that poke out either to the outside of the cell or the inside of the cell. Realize that it's pretty hard to flip this baby around and have this part poking to the inside and this part poking to the outside. To do that, you got to drag this hydrophilic stuff all the way through that big phospholipid bilayer, and vice versa. You got to drag this one all the way through this. So this protein's sort of bobbing along, but it can't really torque around. It's in that fairly fixed orientation such that the protein will have an exterior-facing part and an interior-facing part. On the exterior-facing parts, it's quite common for both plant and animal cells to attach carbohydrates, chains of sugars. Right? In animals, especially animals like you and I, that sugary stuff attached to the proteins makes something called a glycocalyx. It's a sticky, sugary sort of surface that helps cells adhere to other cells. It's literally the glue that holds one of our skin cells to the next of our skin cells, and one of our muscle cells to its neighboring muscle cell. It's that sugary stuff that gets attached to these membrane-bound proteins. And you'll notice, check this guy out. We'll talk more about these kind of things in another lecture or two. This protein has a channel running through its middle. How about that for a neat structure? What do you suppose the functionality you get from a structure like that, where you've got a hollow tube running through the middle of the protein? That's like a gate, isn't it? Whereby molecules might be able to come from outside to inside or from inside to outside. Maybe that's how water molecules do move in and out. Maybe that's how sugar molecules make it from outside to inside. So, but I think most importantly here, those membrane-bound proteins are regulating the passage of things across the membrane. You know, that's the neat thing about a gate. If you have a gatekeeper, you can make sure that some things stay on the outside of the gate and some things get to the inside of the gate. You can be selective in terms of what passes through. You might be able to set up these channels in a way that allows some molecules like sugars to move through, but not other molecules like antibiotics or proteins or whatever. Right? And we'll talk more about those in a little bit later in this class, a separate lecture. So those are that's the nuts and bolts of the cell walls, more importantly, the cell membranes. Prokaryotes and eukaryotes alike have plasma membranes set up in that way. The difference between prokaryotes and eukaryotes is for prokaryotes, that's it. For eukaryotes, not only do we have that bag, but inside of the bag, we've got smaller bags, smaller internal compartments made from the same kind of phospholipid bilayer. So let's start now talking about some of those compartments, like a nucleus. Here's the cell, artist's rendering of eukaryotic animal cell in all of its glory, right? All those things. We're going to start by talking about this guy here, the nucleus. So there's a zoomed in view, peeled away all the rest of the stuff that shows you what this artist thinks a nucleus looks like. The picture is meant to convey a couple of important things. Right out of the gate, though, let me make sure everybody's on the same page. Whereas in prokaryotic cells, a big part of the bag of goo is the chromosome. In eukaryotic cells, the only place you find the chromosome is in the bag, inside of the bag, this bag. So the nucleus, first and foremost, is where it is that eukaryotic organisms store their genetic material. It's where they store their DNA. That's where it hangs out. You don't find stray chromosomes hanging out somewhere else in a eukaryotic cell. They are all clustered in this one compartment that is specialized in terms of storing and using that information in our nucleic acids. So the nucleus, first and foremost, is where chromosomes, where DNA is stored. The DNA that's inside of a eukaryotic cell turns out to not just be naked DNA molecules. In prokaryotes, the DNA that you saw in that electron microscope in the other picture, it's mostly just DNA. There are going to be some proteins associated with it, but it's mostly just DNA. In eukaryotes, it's kind of the other way around. In eukaryotes, 2 to 3 times the mass of a chromosome is protein relative to nucleic acid. So we talk about chromosomes as if they're all DNA. It turns out there's a lot of protein that's glommed on and associated with that DNA to help organize it, compact it, make it manageable, fold it up into nice, neat little packages, as opposed to having these strands tangle with each other and get knotted up. That interaction of protein and DNA together in the nuclei of eukaryotic cells gets a fancy name. It's called chromatin. And so that's what that word chromatin means. They're showing you an arrow pointing to some of these little squiggly things. The squiggly things are nucleic acids, but they're nucleic acids that have a lot of protein associated with them as well. Looking a little bit beyond, what else is this artist trying to draw your attention to? Clearly he thinks these little pimple things are important, right? The little pimple things are nuclear pores. And they're channels through which things, pretty big things, like ribosomal subunits and RNA molecules, can move in and out of the nucleus, right? But they are pores, they are complicated structures made out of proteins, and they're sentries. They're gates, but they're not just an unmanned gate. These are gates where certain molecules are allowed to pass in one direction and not another, and some molecules just can't ever get through in either direction. Right? So that's chromosomes, for instance, just can't ever get out. And then there's one other thing that is really subtle in this particular picture, but to me it's a pretty big deal. Let me see if I can find a good picture of it. See this zoomed in version right around here? And you can see a hint of it over there. You know what that line is? That line is supposed to be a phospholipid bilayer. Okay? And notice what this phospholipid bilayer does. The phospholipid bilayer curls around and it comes back over there. This isn't a phospholipid bilayer in some sense. This is a double phospholipid bilayer. So, prokaryotic cells are a bag of goo. They're a bag where the bag is a one-layer bag, right? The nucleus is double-bagged. The nucleus has two phospholipid bilayers, right? You know what double bagging is. You go, you buy a big watery thing from a sliced watermelon at the supermarket, right? One bag might get broken, it's too heavy, so they put it in two bags. The nucleus is double bagged. It's a double phospholipid bilayer. And that gives it a special name. Oh, well, they're trying to draw your attention to it over there. That gives it a special name. It's not just a nuclear membrane, it's actually a nuclear envelope, because of, to draw attention to the fact that you've got those two phospholipid bilayers. There's an inner phospholipid bilayer and there's an outer phospholipid bilayer. Now you guys probably hadn't had anybody draw your attention to this before, right? The fact that the nucleus has a double membrane, that it's a nuclear envelope as opposed to a nuclear membrane. Why do you suppose it's such a big stinking deal to me, right? It's because I've got this weird sort of warp in my way of thinking about things. I want you to notice as we go through these cell structures for eukaryotes, the nucleus is not the only thing that is double bagged. There are two other structures that are double bagged. And when you look at what's double bagged, what things they have in common, that the other bags inside of the eukaryotic cell don't have, you start to think, how did that happen? How did that get set up? And it gives you some really interesting insights in terms of the origins, the evolutionary origins of eukaryotic cells relative to prokaryotic cells. Right? So the first of these double-bagged things that we're going to be talking about is the nucleus. When we get through the second and the third, then I want to explore with you what the implications might be once you see the things that they have in common. So we got this double bag, the nuclear envelope that we call the nucleus. DNA is on the inside, other stuff is on the outside. Working our way out from the nucleus. And that's a pretty reasonable way to think about a cell, by the way. The limit on the size of a prokaryote, a eukaryotic cell, is ultimately determined by the sphere, the distance over which the DNA of a prokaryote and a eukaryote can sense and control an environment. Right? You can't make a eukaryotic cell much bigger than it is because it would take too long for information to get from the hinterlands, the periphery of the cell, to the nucleus. It would just take too long for that nucleus to exert effective control over the chemistry that's going on at the outer edges. So there are practical limits to how big cells can be. You can't be too far removed. You can't let things get too far removed from that control center of the nucleus. And so again, working our way out from the nucleus to the periphery. The control of the nucleus is greatest right close to it, and it dissipates as you get further away. But as you're working your way out, you'll notice they've got this fancy kind of membrane system that they're showing you in this particular picture. I think the next slide shows an even better picture of it. Let me see if it does. Well, it's showing you a zoomed-in version of it, and then you get out here to this. Let's start with this, and I'll go back to the previous slide in just a minute. So we've gotten rid of the distracting, gaudy chromatin, right? And now we're just left with this outer shell of the nuclear envelope, and you can see how the nuclear envelope is actually integral with this layer of additional membranes that are outside of the nucleus. I got a great word for you guys right now. This is one that you will find you can score enormous success with in social settings where you're trying to impress members of the opposite sex. All right, so this is my gift to you today. Get ready. This is one of the coolest words that you're ever going to get in a biology class. I want to talk with you for a little bit now about the reticuloendothelial system. It just has a nice roll to it, doesn't it? Ah, yeah, boy, you've got a beautiful reticuloendothelial system. Let me tell you, I'm a fan of reticuloendothelial systems. Yours, top notch, right? How can that not work, right? Very effectively. What a fine, I can tell you're a person who possesses just an exceptional reticuloendothelial system, right? The reticuloendothelial system is not a word that you will encounter in casual conversation that often, but that's what's going on right here. It's this layer of membranes that are associated with the nucleus, especially as you're starting to work your way out. Now, while it sounds really cool, it does get to be a bit tiresome. It's cumbersome to keep saying reticuloendothelial, right? And so, a lot of times you'll see it's just abbreviated as ER, right? And they even flip it around. This is the endoplasmic reticulum, okay? But it is the reticuloendothelial system to those who are sophisticated and in the know. But often you will have been exposed to this concept in the past as the endoplasmic reticulum. And even that's kind of cumbersome, so they abbreviate that as the ER. The endoplasmic reticulum is often broken up into two different sorts of flavors. There's the rough ER, which is typically closest to the nuclear envelope, and then further out is the smooth ER. Here it's shown as being more spindly, more dissipated, further removed. The big difference between the rough ER and the smooth ER is what makes the rough ER rough. With a microscope, a really good microscope, probably just with an electron microscope, what you see in the rough ER that makes it rough is embedded in the endoplasmic reticulum are these studs. They look like little studs, which are ribosomes. Ribosomes are pretty big complexes of proteins. They're things that you could conceivably see with a light microscope. That's a pretty big assemblage of proteins if a light microscope can let you see something at the level of a protein. The rough endoplasmic reticulum is rough because ribosomes are physically embedded in that phospholipid bilayer. I haven't said it very plainly yet. I think I've mentioned it, but ribosomes are where proteins get born. Ribosomes take information from the nucleus in the form of RNA molecules, and those RNA molecules get translated into proteins. The thing that does the translating are ribosomes. There are two kinds of ribosomes. There are membrane-bound ribosomes, those in the rough endoplasmic reticulum, and there are free-floating ribosomes, the ones that are just floating around in the goo of a eukaryotic cell. But what makes the rough endoplasmic reticulum rough is there are ribosomes embedded in it. You now know ribosomes are what makes proteins, if you didn't already. What do you suppose is going on in the rough endoplasmic reticulum? Proteins are getting made. And not just any kind of proteins, two specific kind of proteins that need to be membrane bound themselves, like the ones from, 4 or 5 slides ago, and proteins that need to be secreted from the cell, dumped off, or delivered to some other compartment inside of the cell. Okay? So that's what makes the rough ER special, is ribosomes are there, and those ribosomes have the job of making membrane-bound proteins as well as proteins that need, especially the proteins that need to be secreted from a cell. As you work your way out through the endoplasmic reticulum, and now I think I can go back. So here, look at this. Here's that zoomed in version. This grainy black and white image, that's code for an electron micrograph, a transmission electron micrograph image. So here's a real zoomed in version of the rough ER, and you can see what's going on. You got those studs there, doesn't that? What's a better word to describe this? Is this a smooth surface or a rough surface? Hence, rough endoplasmic reticulum. You can see those membranes are have these studs, these black specks, these ribosomes associated with them. Ribosomes are these big guys. We'll talk more about them a little bit later in the class. They've got a large subunit and a small subunit, but they're hanging out here in that rough ER. So there's the nucleus. Working your way out, you can see they're showing you the rough ER. Here's a zoomed in version of the rough ER. Here's the rough ER, but now you're also starting to see the smooth ER, which comes along a little bit later. Now, I made a big deal about the reticuloendothelial system, right? And I think that is kind of important in the sense that I want you guys to appreciate that, sure, people talk about the rough ER, and that's where proteins get made, right? Membrane-bound proteins in particular. And then people talk about the smooth ER, and that's where the proteins that got made in the rough ER get processed, sugars get tacked onto them, for instance, stuff like that. It's also where a lot of membrane synthesis occurs. That's where the phospholipid bilayers get made so that you can make bigger membranes. That's the smooth ER. Just out from the smooth ER is another thing that's physically attached. It's called the Golgi apparatus. All three of those things together are the reticuloendothelial system. And while it's convenient for people to talk about a rough ER as being distinct from a smooth ER, there is no bright line that you cross from the perspective of a cell. They just blend together, right? There's no sharp transition. Oh, I'm not in the rough ER anymore. This is smooth ER territory. It's just like, gee, you're not really sure when you've crossed over the boundary from rough to smooth ER as you're working your way through the reticuloendothelial system. So it's all one big package from the perspective of a cell. At one end of the continuum of the reticuloendothelial system, there's a region that has a lot of ribosomes. At the other end, there's a region that just doesn't have any, right? But it's just sort of this gradient going from the beginning of the reticuloendothelial system to the end. So here's that third part of that reticuloendothelial system. They're trying to show you now, focus your attention on the Golgi apparatus. And here in the Golgi apparatus, you can see that little buds or vesicles of the Golgi apparatus can pinch off, and you get these little internal membrane-bound compartments that sometimes have stuff in them, like proteins that were made in the rough ER, and those confuse with the cell's outer plasma membrane, and in doing so let go to the outside the stuff that was encapsulated inside of the membrane. So altogether, those three things are the reticuloendothelial system. Right? We got your endoplasmic reticulum as well as the Golgi apparatus. And I tell you what, hey, you know, I've got a couple more things that I can talk with you about today. Clearly, I'm not going to get through the whole outline during the course of this lecture. And I'm mindful of the fact that at the end of the semester, you guys are going to fill out student evaluations about how effective an instructor I've been. I want you to remember when you fill out those evaluations that I didn't always keep you the entire length of the class. So why don't we stop right here today, 5 minutes early, my gift to you, and we'll pick up talking about lysosomes at the beginning of class on Thursday. All right? See you guys then.